In this lecture, we're going to finish up the rest of the material that I'm directing you toward in the notes package. Again, like the previous lecture, if I've got a lot of information available to you in the notes package, then I am not going to spend a lot of time describing that. We're going to just focus on the blanks, I guess, in the notes package that I've given to you. All right, so let's get started and talk a little bit. So we're starting here in the notes package where we talk about the conduction of action potentials. So in an unmyelinated axon, the entire membrane of the axon must undergo action potentials. And so one action potential that is traveling down the membrane will, so if an action potential occurs in one section of the axon membrane, then, so if this is the axon, so an action potential occurring in this zone of the axon will then trigger or stimulate an action potential to occur in the next zone, which then triggers the next zone, and so on. But if the axon has a myelin sheath around it, then the myelin acts as an insulator, and that region of the myelinated region of the axon will not undergo an action potential. The only area that undergoes the action potential is going to be this area here, the node of Ranvier. And so this region will undergo an action potential. This region is insulated, and so that action potential is not required, but we do see really rapid conduction of the signal across the myelinated area, and then the next zone or the next node will undergo an action potential. So this propagation pattern here, where it skips, where only the nodes are undergoing an action potential, is referred to as saltatory conduction. Because saltation is a type of movement that involves leaps or jumps, and because the action potential appears to jump from node to node, then what we're seeing is something that resembles this kind of saltatory or jumping kind of movement. So what factors can influence the rate, the speed with which impulses are conducted? And there are three factors that can influence the speed or the rate of impulse conduction. First, larger diameter axons will allow for faster conduction of the action potential. And we saw this in the interactive physiology in that presentation as well. And it's because resistance to the electrical resistance decreases as diameter increases. And so what we see is that many of the sensory neurons, and including touch, pressure, proprioceptors or joint position sensory neurons, and even some of the temperature sensing neurons, all have really large axons. And so this helps to speed the conduction of the nerve impulse. As you might guess from our conversation about saltatory conduction, if an axon is myelinated, then because only the node is required to undergo an action potential, and that signal or the depolarization is rapidly spread across or along the membrane that is wrapped up and covered by the myelin sheath, then this also speeds impulse transmission. And this is also a more efficient form of, or more efficient process, because we're moving less sodium. Only sodium and potassium at the nodes of Ranvier are moved. Then it doesn't require as much ATP for the neuron to restore resting membrane potential. And last of all, if a region is cooled, then the nerve impulses are going to be conducted more slowly. And this is one way that icing, or this is how icing helps to decrease the amount of pain that one feels when one injures oneself, because we conduct the pain signals much more slowly toward the brain. And that slower conduction and the slower arrival of action potentials at the brain, that is a signal that there's less pain being produced. Okay, so what else, or how else do we describe the rate of, or what other factors might affect the rate of impulse production? Well, the amount of stimulation that's being received. So, for instance, the amount of pain that we might feel, or the amount of pressure that we feel, these sensations are more intense if more action potentials are being sent. So the thing that I want you to get from this is that if we're feeling more pain, let's say, or if we're getting a lot of bright sunlight coming in through the eye, we register that sensation, or the intensity of that sensation, not because really strong action potentials are being sent. And so that's a point that I want you to get. There's no such thing as a strong action potential, because every action potential has the same intensity. It's got the same amplitude and the same length of time. It has the same duration. The way that we register a really strong piece of sensory input is the number of action potentials that are sent in the same amount of time. We increase frequency of action potentials, and that tells us we're receiving really intense sensory information being distributed. So the difference between just a little bit of pain and a lot of pain is how rapidly the brain is receiving pain impulses, not because we're sending really strong action potentials. Okay, so let's talk a little bit about synapses. These are the junctions across which impulses are transmitted, and they're transmitted using neurotransmitters. These are chemicals that are produced and then secreted by the presynaptic neuron. And so we talk about this kind of synapse as a chemical synapse, because it's chemicals, because it's chemicals that are producing the effect on the postsynaptic cell. So the axon terminal, that's the knob-like ending there of the presynaptic neuron. That's where the axon comes to an end, and it is the axon terminal that's packed with synaptic vesicles, and it's the vesicles that are then filled with neurotransmitter. There's the synaptic cleft, as we're looking at the anatomy. That's that space. It's a very, very, very tiny space, 20 to 30 nanometers. That's what this NM means. A nanometer is a millionth of a meter, and the neurotransmitter has to diffuse across this cleft. And then finally, there's the receptive region. That is the membrane of the postsynaptic neuron, and it's going to have lots and lots of those receptor proteins that are going to open sodium channels or other kinds of channels as well, depending on what kind of signal is being produced. But as we open these sodium channels, that's going to cause a local depolarization of the receptive region of the postsynaptic neuron. So how do we send information across this this synapse? What are the steps that are required? And as you go about trying to explain storyboarding your the information about how a an action potential is being sent, I want you to also include how we get that signal across the synapse as well. So when an action potential arrives at the axon terminal of the presynaptic neuron, when it reaches that synaptic bulb, that opens voltage-regulated calcium channels. So the calcium channels open, and calcium rushes into the axon terminal. Now we saw this same effect at the neuromuscular junction. With calcium rushing in, then that triggers the exocytosis, or the movement of the synaptic vesicles that are containing the neurotransmitter. It causes them to move to the membrane there at the axon terminal to exocytose then the neurotransmitter that they contain into the synaptic cleft. Now those empty vesicles move back to the cytoplasm of the axon, and then they'll be refilled there, and other full vesicles will move into their place, and the calcium ions are going to be quickly moved from the cytoplasm by active transport. So we'll use ATP to gather up calcium inside the postsynaptic or the presynaptic neuron there at the axon terminal, and so that calcium is then rounded up and very quickly moved back out into the extracellular fluid. Okay, so we've got then the axon terminals now reset and ready to receive a new action potential. What's happening to the neurotransmitter that we released into the synapse or into the synaptic cleft? Well, it diffuses. So diffusion is the method by which it moves across the synaptic cleft, and there it does this temporary binding to receptor proteins. Now, when a neurotransmitter binds the receptor protein, it causes the proteins to change shape, and as they are changing shape, then they open ion channels in the postsynaptic neuron's membrane, in the receptive region there. And so depending on the ion channel that opens, then we're going to see different effects, and depending on what neurotransmitter is released into the synapse, that determines which of the ion channels open. And so ions then will flow into the postsynaptic neuron, so across that postsynaptic neuron's membrane, and that generates a local potential change. And that local potential change will then spread across and down to the cell body of the waiting neuron. If sufficient stimulus is received at the axon hillock of the postsynaptic neuron, then an action potential might be generated on the next, or then it will be generated. If there is sufficient stimulation, then it will generate an action potential down the axon of the postsynaptic neuron. But it all depends on the kind of stimulus that's received back here at the dendrite of, or even directly on the cell body of our postsynaptic neuron, because it can receive either excitatory or inhibitory stimulus. So if it is excitatory, then we open sodium and potassium channels. We'll open, and we will see the effects really similar to what we saw with an action potential being spread down down the axon. We're going to see the sodium rush in, the potassium rush out, and that will depolarize the membrane, bring the membrane of our cell body a little bit closer to reaching threshold. So depolarization, remember, that's a positive change in membrane potential. So the membrane becomes less negative, gets a little closer to that minus 55, moving up from minus 70 toward minus 55. But, sorry, Let's go back there. But sometimes, or depending on the neurotransmitters that are being sent across, the potential change may be an inhibitory one. This is one that causes hyperpolarization, that decreases the membrane potential. And so it does this by opening potassium channels, so potassium leaves the cell, or it can open chlorine channels. So there are channels that move chlorine. And if chlorine rushes into the cell, that will make the cell more negative. If potassium rushes out of the cell, that will make the interior of the cell more negative. And that reduces the chance, because we're making the interior more negative. We're taking, it's at minus 70, and we're making it even more negative, maybe bringing it to minus 75 or minus 80. That makes it harder for the axon hillock to come to threshold, because we're moving the wrong direction with the membrane potential. So excitatory neurotransmitters are going to bring us closer to reaching threshold, while inhibitory neurotransmitters, their effect will bring us further from that threshold potential, membrane potential. So all of those steps, from the calcium entering into the presynaptic neuron, causing the synaptic vesicles to come and disgorge their neurotransmitters, to the neurotransmitter diffusing across and opening those, whatever ion channels it opens, depending on whether it's excitatory or inhibitory. All of that takes between 0.3 and 5 milliseconds. Super short time period, but it's a measurable time period. And we talk about that brief amount of time as synaptic delay. And so as long as we have neurotransmitter molecules in the synaptic cleft, then, so this is beyond the synaptic, beyond that 0.3 to 5 millisecond delay time, then as long as those neurotransmitters are there in the synaptic cleft, they will continue to hold the ion channels open. And so in order to end the stimulation that the postsynaptic cell is receiving, we've got to remove the neurotransmitters. So there are a couple of different ways that we can remove neurotransmitters. The first is that there are maybe enzymes on the the postsynaptic cell membrane that will digest and break up or cut up the neurotransmitter. So for instance, we've already seen at the neuromuscular junction that the enzyme acetylcholinesterase will digest or break up acetylcholine, and then the pieces and parts are moved back into the axon to be reassembled. So there's a second technique called reuptake. And what reuptake does is to take that neurotransmitter whole back into the presynaptic neuron. And so rather than breaking up the neurotransmitter like we saw with acetylcholinesterase, instead we just grab the neurotransmitter and do endocytosis with that neurotransmitter and reform vesicles filled with neurotransmitter. And they can be released again, or in some cells, rather than being gathered up and released once more, then enzymes inside of the presynaptic neuron will digest the neurotransmitters. So for instance, norepinephrine is reuptake occurs of norepinephrine when we release it as a neurotransmitter into the synaptic cleft, and then it will be immediately reused, while biogenic amines and amino acids that act also as neurotransmitters, they are taken back up by endocytosis and then broken down by monoamine oxidase, which is an enzyme that will break up these amino acid and the biogenic amine, those neurotransmitters. And there are some kinds of depression treatments that work by inhibiting monoamine oxidase. They're called MAOIs because they inhibit monoamine oxidase, and they help to prevent the destruction of the neurotransmitter so that more neurotransmitter is available to be released again when an action potential arrives there at the axon terminal. And then there are also reuptake inhibitors that will help to ensure that reuptake doesn't occur. And so with some of the drugs that we use to treat depression inhibit the reuptake of serotonin. They're called selective serotonin reuptake inhibitors, SSRIs. So SSRIs and MAOIs help to fight or treat depression or other conditions like anxiety by forcing the neurotransmitters to remain longer in the synapse. Then last of all, with some neurotransmitters, they just diffuse away from the synapse and out into the tissue fluids, where they will be neutralized by enzymes out there in the tissue fluids. So sometimes there's just not anything. These are neurotransmitters then that take a little bit longer to be cleared from the synaptic cleft than those that are destroyed by enzymes or reuptaken. I'm not sure exactly how to say that. All right, so then I give you a whole bunch of information about synaptic potentials, the excitatory and inhibitory postsynaptic potentials. I'll let you read that on your own. And we're going to move on to talking about neurotransmitters. Neurotransmitters are a group of chemicals that are produced by neurons and then released into the synapse to help either inhibit or excite the postsynaptic neuron. There are about 50 chemicals that are suspected of being, that are known to be neurotransmitters, and another 50 or so chemicals that are suspected of being neurotransmitters. And there is a table in the book, I'm not sure right now, I don't have my book open, but look for the table that lists out many of the known neurotransmitters. So most neurons, they produce only a single neurotransmitters, but there are some that can produce two or more even neurotransmitters, and they are capable of releasing all of the neurotransmitters, or not all of the different kinds of neurotransmitters at one time, or to selectively release only one of those or only two of the neurotransmitters, perhaps, that they produce. And when we look at how we classify neurotransmitters, they can be classified either based on their chemical structure or the function that they serve. So let's first look at the chemical categories. So we're looking at the chemical categories of neurotransmitters. Acetylcholine is one of the neurotransmitter types, and so many, well, we've already talked about the fact that neuromuscular junctions are run, or the neurotransmitter that we use there, the neuromuscular junction, is acetylcholine. It was the first neurotransmitter to be identified. Back in 1921, we identified that. And the way it's removed from the synapse is through the activity of an enzyme, acetylcholinesterase. And what we see is that this is an excitatory neurotransmitter when skeletal muscles are involved. So it helps to bring a skeletal muscle a little bit closer. One shot of acetylcholine will help to bring a skeletal muscle a little bit closer to an action potential, while at cardiac muscle, acetylcholine actually acts as an inhibitor to bringing the cardiac muscle closer to the threshold for having a contraction. So acetylcholine is one of the chemical categories, and there's only one here. A lot of the nerve gases that are used in warfare and organic or those organophosphate insecticides, like we talked about in class yesterday, they achieve their effect by inhibiting acetylcholinesterase, and that ensures that acetylcholine remains active in the synapse. Biogenic amines, also called monoamines, because only one amino acid is found here. These are neurotransmitters that have been synthesized from one of three amino acids: tyrosine, tryptophan, or histidine. Those are each an amino acid, and so we produce biogenic amines from one of these three amino acids. Depending on the cell that is involved here, depending on the postsynaptic cell, then these biogenic amines produced from these three amino acids, their release may have excitatory effects or inhibitory effects. It really depends on the receptor that is binding them. But we can cluster biogenic amines into two different classes. There are the catecholamines, and these include dopamine, norepinephrine, and epinephrine. And so, we haven't talked much about epi and norepinephrine, but these are hormones, but they also can act as neurotransmitters. So depending on the location, the presynaptic cell may produce norepinephrine or epinephrine and use that as a neurotransmitter. The indolamines include serotonin and histamine. So we've already talked about histamine as being a product of a mast cell, but histamine can also be used as a neurotransmitter between two different, a neuron and another neuron, or some other tissue. So catecholamines or dopamine, sometimes thought of as like one of the happy drugs, because dopamine release in the brain generally promotes feelings of euphoria. Norepinephrine and epinephrine. Serotonin is one of the drugs that we one of the neurotransmitters that we tend to be messing around with when we're trying to treat depression, trying to help serotonin remain in synapses. So you may have heard of that before. And histamine. Those are both indolamines. What else to say here? The MAO, monoamine in oxidase, is used to break down these biogenic amines. Okay, a third category, chemical category, are amino acid neurotransmitters, and so there's a number of them. Gamma aminobutyric acid, also called GABA, is the most common of the inhibitory neurotransmitters that we find in the brain. So a lot of anti-anxiety drugs, Valium, for instance, are GABA agonists. That is, they enhance the action. They enhance this inhibitory action of GABA. Glycine is another inhibitory neurotransmitter, and it is very common in the spinal cord. So GABA is a inhibitory neurotransmitter in the brain, glycine inhibitory in the spinal cord, and aspartate and glutamate, these are two excitatory amino acids. Glutamate is responsible for perhaps as much as 75% of the excitatory transmission that we see in the brain. So amino acids they play a huge role also as neurotransmitters. So they don't just make up proteins, but we use them as neurotransmitters as well. And then little short chains of amino acids, peptides, sometimes called neuropeptides because they're acting in the nervous system. These peptide neurotransmitters may be chains of 2 so just a dipeptide, or as many as 40 amino acids. So these are like tiny little proteins. And they're found both in the central nervous system and the peripheral nervous system. And frequently, these peptide neurotransmitters act as something called a neuromodulator. So they release and they help to regulate the effect of other neurotransmitters. So they're released alongside these other neurotransmitters that they're regulating, and they can either turn on or turn off the neurotransmitter, the action of other neurotransmitters. So that's what we mean when we call them neuromodulators, because they help to regulate other neurotransmitters' activity. Okay, so those are the chemical classifications, and then we can classify neurotransmitters functionally, either as excitatory or inhibitory. So if it's excitatory, if it's an excitatory effect, then it changes the resting membrane potential, making it less negative and bringing it closer to threshold. So this would help to stimulate a response at that postsynaptic cell. If it's inhibitory transmission, if it's a neurotransmitter that results in inhibition at the postsynaptic cell, then it's making the resting membrane potential, bringing it even lower. So we're going from minus 70 to minus 75, oops, sorry about that, or even lower, to make it harder for the postsynaptic cell to reach threshold. There are those that have ionotropic and metabotropic effects as well. Ionotropic neurotransmitters, they open ion channels, and this causes immediate changes in the resting membrane potential as we see ions moving from one side of the neuron membrane to the other. Or it could have a metabotropic effect. That is, it activates some metabolic process in the postsynaptic cell. So it's not only always about bringing the cell to threshold, it could be, this neurotransmission could be about activating processes or inactivating processes in the postsynaptic cell. So that would be a metabolism acting or a metabotropic effect caused by a neurotransmitter. Okay, almost done here. Let's talk last of all about neuron circuits. So neurons tend to function in groups with, and those groups kind of all, or that group of neurons, these are neurons that are all working together to be involved in a common function. And so we talk about these common acting neurons as forming a neuron pool or a neuronal pool. So neuronal pools, they can have thousands to millions of interneurons. So neuronal pools are found in the central nervous system, where we're trying to either minimize the effects by getting fewer and fewer neurons involved, or maximize the effect by activating more and more neurons. That's what these neuronal pools can do. And so a neuronal pool is found among neurons that are receiving, processing, and then sending on information to groups of or links of neurons. So the simplest neuronal pool might be might just involve two neurons, an incoming neuron and the neuron taking the impulse away. So these neuronal pools then are just groups of neurons that are working together. So we can see two different types of circuits within a neuronal pool. One is called a converging circuit, which is a neuronal pools or pathways in which we are concentrating the transmission onto fewer and fewer neurons. So here we see a very simple converging circuit where we see two neurons that are focusing their axons and their axons transmission onto a single cell, so that we're concentrating this signal toward one individual cell or a small group of individual cells. These are converging circuits or converging pathways. And so, for instance, we might see converging circuits working when we are trying to maintain balance in the body. So that requires integration of input from the eyes, from the inner ears, and from stretch receptors in the neck that are all funneled toward a relatively few number of neurons in the cerebellum. And it's the job then of the cerebellar neurons to be able to integrate all of this differing kind of sensory transmission into one coherent message and respond to that. And so that helps us to maintain our sense of balance. So the opposite of a converging circuit then would be a diverging circuit, where one or a small group of neurons are sending axonal transmission to one neuron and then to another neuron via a collateral. And so one neuron is then spreading its signal to multiple neurons, and these neurons then may be spreading out their signal to even more neurons. These are diverging pathways. And in these kinds of circuits, what we see is that one neuron may produce output on dozens of neurons. So we get those effects seen in multiple places. All right, so there it is, convergent and divergent circuits. As always, if you have any questions, be sure to contact me. This is all that I've got to share with you about this chapter. Study well, and we'll see you on Monday. So remember, we won't be having class on Wednesday next week, but we'll see you on Monday of next week for the exam.